I think this is probably gonna be it for today. People are already celebrating Halloween, which is nice. Glasses to Halloween. Well, you guys deserved it, because you guys kicked that midterm butt. I was super nice, I was super excited to grade your stuff, and I was like, A's everywhere. No, you guys did a really good job. The average again was a 90. Then my Excel sheet kind of keeps track of like your midterm versus your lab midterm. It'll give me like your trends. I saw a lot of people really stay within 5%, like your grade didn't drop more than 5%, which is good. So that was a lot of them. There was a few that I saw that there was like more than 5 or 7% increase in your grade. So if you did that, you know what I mean, you're improving. I really like that. There was only there was a few cases where the grades came down a little bit, more than 5%. So if you throws me like a little red flag telling me, hey, your grade dropped a little bit from last midterm. So if you get an email from me telling you, hey, I want to talk to you, don't be super like, it's not maybe just due to that. It tells me like, hey, watch out, like somebody's grade dropped. So most of them, they were still really high, like you're still in the being an A range, so it's not like, I'm not like, oh, I'm not too worried, but it's like, hey, something happened, it's okay, but you know, keep up the hard work you were showing me before. Can't find my notes. This class is Smith, right? So it's gonna be a pretty fast class. We're gonna be talking about the membrane. And I don't think I have big announcements other than everyone submitted their paper, their essay so far? Yeah? If not, get it to me, please. That way I can start grading those over the weekend. 25 points, so make sure you guys do it. And then next week, I think next week you have an in-class assignment, right? I feel like it's in-class. So we don't have homework, so I'm not telling you anything about that. And what else? So we're getting down to the last midterm, the last finals coming up. That one's worth 75 points, it's a little bit more. So you're gonna have a little bit of stuff that's gonna kind of follow through you that we started talking from the beginning all the way to the end. It's gonna have a little bit more there. I'm gonna start making the section 3. I already made it. It should be in that first folder. That review thing for midterm number 2. I think only like one person contributed to it, like one slide, and then that was pretty much it. So I'm liking this idea of during the midterm, I give you a word bank. And I want you to use all the terms in that word, like for the last page, right? When I was like, hey, tell me the story about transcription. And I give you all those words for you to use. I'm thinking of making a word bank in that review thing, so that you guys can study them. I do have notes, so I'm just gonna put the word. So you just know that at the end, for the final, when I ask you to tell me a story about something, I'm gonna give you a word bank that I want you to pick from it and pick multiple terms to tell me a story. So it's gonna be more of an open-ended question. So it might be a little bit tougher on you guys. But the thing that I've been liking is when I gave you the homework last time, I told you to come up with questions on your own. You guys are coming up with good questions. So it means that you guys can actually think about the material, come up with a strategy or something to quiz it. In the final, if I decide to tell you, here's a word bank, you tell me a story about how these things work together or how these things are like different levels of life come together to give this bigger effect of how life happens. I feel like I'll feel confident that you'll be able to write it. Yeah. So it's gonna be, I don't know, it might be tougher or easier for you, but that's kind of what I'm thinking right now. So having a key bank, a word bank will help you. If you don't want to do it with the one I have in the class, fine, make your own, right? Add your notes, add pictures, whatever you need to make it more understandable to you. Yeah, so with that being said, on to part number 3, guys, we're almost done, woo! All right, let's start talking about the cell membrane today. Perfect. All right. So cell membrane, you guys remember that I always talk about it, super simplified. I'm gonna say, okay, cells are basically like a little fat bubble. So this, I'm happy you guys have to go back to your notes and bring this up again. So that you have the phospholipid bilayer, right? So we're gonna have to bring that up all the way back up. So here we have this really cool picture. Here you have your phospholipid bilayer, right? Up here, you have our phosphate groups, and here you have your tails. You guys remember this part? We're gonna go back to these really simple drawings like that, right? On this side, we have our this side would be facing, let's say, our cytosol, right? So we're looking at the inside of the cell. On this part, we're looking at the outside, which is the exterior, right? So you guys remember this part, these molecules, they have different and special groups. On the inside, we have these chains, which is carbons and hydrogens, right? Which is kind of what we're showing here. Because they're carbons and hydrogens, we're gonna go back to my old favorite words of hydrophobic, hydrophilic, right? So, guys, a little flashback, remember, hydrophobic, the lover is scared of water. Scared. That's right, scared. Opposite side of that, we have here the drawn in blue. That's your hydrophilic side. What does that mean?
Interaction with water,
I think I heard it, somebody.
loves water, interacts with water.
Interaction with water, perfect. So the whole thing of this being held together is one is that in the exterior, in the cytosol, we have a lot of water, right? That means that when it's time to interact, the side that loves the water is gonna interact with the water. The sides that hate the water are gonna basically curve into themselves, right? One thing we're gonna talk about later on, or when we go deeper into the presentation, is about how on the inside, what's actually holding this together, right? One of them is you have the water molecules interacting with the outer parts, which don't let these molecules basically swing out like this. Because if one of these try to open and go this way, you have a hydrophobic part trying to interact with water, it's not gonna be happy, right? The other one is hydrophobic molecules can interact with other hydrophobic molecules. Remember those really weak van der Waals interactions, or we have a little bit of those small, basically like the net charges that come loose slightly from one to the other one, and they basically like glue themselves for like half an instant, like it's super weak. With these double carbon chains, what you're having there is you're having a lot of the interaction happening, right? So on the inside, you have the forces of them interacting with each other, kind of hold each other in place. And at the same time, you have the water on the outside, not going to basically come up or interact. All right. So when we talk about the membrane, we're going to go deep into that today. That's my little quick review. And then we're also going to go into the proteins that are found there. So hopefully you guys have been doing the reading. Yes, 7.1, 7.2. Yeah, I see one. Thank you, sir. So for this one, we're going to be talking about the fact that we do have this phospholipid bilayer. Here I'm drawing it kind of complicated. Later on, you can see we essentially draw two lines. We'll talk about how they interact. Within that, what you're going to find is it is a complex structure itself. They didn't put that much into the book. They talked a little bit about these lipid graphs, but they talked, like I've heard, or the work I've seen before when they talked about it was that they talked about how the graphs themselves, you can have different fats that are found here, like your polyata, you can have saturated fats or unsaturated fats based on the carbon chain being like a straight linked up, or they have some kinks that kind of fold down. The other thing they talked about is the length of these molecules can actually be different. Some molecules can have longer tails and shorter tails. And just so those molecules can be longer or shorter, you can imagine that the width of this domain can be affected like this. Right. So you can have a part of this where it's narrow, and if these molecules are longer, you have regions where actually it can be wider, right? So this thing isn't just like, oh, it's 2 layers, exact width, it's perfect all out. It can actually vary. The other thing that can vary is when you go inside of it, you're going to find that there's going to be proteins that are actually embedded within it. For this one, the book talks about 2 different types. I normally think about it like there's 3 different types, right? So some of them, they're going to be your peripheral proteins. And the big thing about these guys is these proteins are interacting. They could be interacting with other proteins, or they could be interacting with the components near the edge of the cell, but they won't be embedded into the phospholipid bilayer, right? Like these would be on the surface. The second 2 are part of the integral ones, and there we're going to be talking about proteins that go into this phospholipid bilayer. I want you to think about it going into the hydrophobic region. And then for that, so there's 2 types. There's one, which would be transmembrane, which means they go all the way from the outside, across the hydrophobic region, all the way to the other side. So like inside, all the way to the outside, while some of the other integral ones, they only sit on one side, they don't make it all the way to the other side, right? And these proteins will kind of be embedded in that tissue. And then we're gonna go into how that's actually functioning. All right, so again, bringing on that big theory or the big topic of the fact that you have a phospholipid bilayer, but it's actually very movable. It's not a fixed structure. It's not something that they're glued next to each other and these molecules can't move. They're actually just kind of sitting next to each other and they have the ability to migrate. So if we imagine for a second that as a class, the desks in here would be your phospholipid bilayer, right? They have little wheels. Let's say these top parts are your hydrophilic regions. These desks, technically, they can move. Sorry. Technically, the desks can move around, right? They'll be shifting around the classroom. They can move this way, right? So within that same layer, these molecules can move around. They're not permanently glued, right? We don't have any covalent bonds. What we're talking about here is just like van der Waal, the fact that they don't want to touch water. Same thing's happening on the other side. Then they can move around like this. The way they can't move around is because they have this hydrophobic region, you can't actually flip them to the other side, right? If you try to flip them, you imagine this hydrophilic region is going to try to interact with this hydrophobic thing, and you know they're not going to be happy. So they can move within their own plane. They're not going to switch over to the other side. And then, like I said before, inside of this, what we're going to find is there going to be multiple proteins. Some of them sitting on the outside, some of them sitting within one of the sides, or going across both of them, right? And then, like I'm saying, these things can move around a lot. Let's go to the next part. All right, so I got a little bit ahead of myself. So now let's go back. Let's zoom into what this is. So here we have one new keyword, amphipathic, right? And what this means is that you basically have a molecule that has two different components to it, right? So this really zooms in and defines these two features. And what you have is you have a hydrophobic region, like again, this is the inside part right here. Then you have your hydrophilic, which is the outer part, which is the water-loving side. And this is a really good picture so we can talk about how they move. Again, because these regions can interact with each other, they can move in this plane. It'd be okay, there's nothing's gonna interact. And they can move this way, but we can't have this happen because of the hydrophilic side trying to come into the hydrophobic region. All right, so here's a little drawing kind of showing you this little diagram. You can see the purple molecules represent those hydrophilic heads, and then they can migrate within that top region. But the thing that you're not going to find, it's going to be hard to find, is for this thing to be able to flip over to the side. So the green side, which is like saying the inside of the cell, will stay on the inside, and the outside will stay on the outside. I think I'm gonna have to update the slide for this one. In the book, they had a really cool one that they went back and they looked at the ER. So you guys remember the ER has two jobs. The rough one makes our proteins, the smooth one makes our lipids, right? So it actually helps make these structures. The cool thing they were kind of showing there was that in the ER, when it blends off and it produces it, they were keeping track of these two sides, right? And what they were showing was that, let's say the purple represents the outside of the cell, right? In the ER, you're gonna have something like this. Right. So you're gonna have your structure looking like that. This is one phospholipid bilayer. This is one side of the phospholipid bilayer. That would be the other side. When this is getting turned into a vesicle, this is gonna get kind of puffed up, right? It's gonna look something like this. Unless you. No, I think I got it. Okay, so we have this. It's gonna be into a vesicle, right? With that, we're getting it to travel through the Golgi. Now we're getting all the way to the surface of the cell. When we get out here, this is gonna go and merge into it, right? So this would be the inside of the cell. And this would be the outside, right? Once we get it here, this vesicle is gonna get nearby. It'll try to fuse itself into this part. This is where the green would look like this. And the purple, right here you have the phospholipid bilayer like that. This is gonna link up with these, and you're gonna basically see that. And you can see that basically the inside of the ER, we're reducing the lipids and we're making this chain be built up. When we travel it outside, that's the one that's gonna give us the outer flap on the phospholipid bilayer. In the book, they have the picture, I forgot to put it in the slides, but I'll throw it on this one on the side right here, for you guys to take a look. You know, in case I want to hear one of those long story ones, that's one I can bring up about the lipids. All right, so talking about this lipid bilayer, it's one of the things that we can talk about is about the fact that it's, again, it's fluid, it's not a permanent structure, it's not perfectly glued to itself. So because of this, we can have this kind of things happen, right? So here you can see that we can basically pinch off this molecule as long as we glue it back to itself, like this, where you have the phospholipid bilayer stable phospholipid, it'll be fine. Here, when we move it to another surface, we can actually glue it into it, and it'll build into this to build a single layer when it's done. And that's kind of what they were trying to look, we're trying to test in this experiment, right? And here, they basically tag proteins on the surface of either mice cells and human cells, and then the orange kind of represents like a vesicle, so it'd be like a fat bubble, right? We have that phospholipid bilayer just like this. They then threw them together, and you got those membranes to fuse, right? So it means that those two fat bubbles came together, boom, give you a bigger bubble. Now that you have the bigger bubble, what they were doing is they were keeping track of these proteins. I forgot how they tagged them, but probably nowadays someone's probably done this with GFP to make them look pretty, but after only an hour, they were actually able to pick this up, right? What they just picked up was that those proteins that were in the membrane, initially you imagine these two come together, they would merge, and you would think, oh, if they're not moving around, half of it should have been blue, half of it should have been red, right? But the fact is that the membrane can move around, the proteins across the membrane can move, so they fuse across each other. After an hour, they able to pick up something like this, right? That means that if something was in this membrane, like a protein, technically it has the ability to move to different spots, kind of like moving around, right? So even if you drop this vesicle from the inside, brought it to the surface, drop its content outside, if you had brought it with a bunch of protein receptors, those protein receptors would then show up at the surface and they could technically start moving and then cover the surface with your protein. All right, so now we're gonna zoom in again, zoom in more, we're going back further into this. We're looking at how the tails can actually help change its properties. So in here, I kind of want you thinking about, it's the size of the tail and the saturation of the tail, right? So the size of the tail is, again, how many carbons you picked up. So this yellow thing, if we super zoomed in, you expect to find something like this. Carbon, 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 carbon to be happy. If you guys remember, it likes to have 4 bonds. In the chain, it currently has 2 other carbons. So that means that it still has these other 2 that are going to be binding with hydrogen. Right? When carbons are arranged with 4 partners, when each carbon has 4 partners, it'll do that zigzaggy pattern and it'll be consistent. So from top to bottom, you'd expect it to be like this. Right? So this, normally in my head, I kind of remember, it's like when they say saturated, it's saturated with hydrogens, right? So you see every carbon has those 2 hydrogens, the last one has all 3. This ends up meaning that when you have this molecule set up, it actually makes a straight line looking something like this, right? So this is your saturated. The other way to look at this is, the other fats are your unsaturated fats. Right? And the things that you're going to find here is when you have your carbon, hydrogen, but you actually have a double bond to the next carbon. In this case, if you guys remember, it does that funny thing where I was like, it binds like this, right? Like it'll do 2 going this way, 2 going this way, but it gives you different degrees. Those degrees give us this pattern. When you have a double bond, it's only gonna have 3 different partners. So what ends up happening is this one sets up, so it's on the same plane, but they're equally distanced like this from each other. And what's gonna happen here is your hydrogen is gonna go there, your carbon is gonna go here, like this. So imagine there was 2 carbon chains above this, they would have been coming straight down at the moment you hit the spot where there's a double bonded carbon, because now this one can only use one more hydrogen, it'll set up in this triangle pattern and it'll bend out to one of the sides. And that's when you have these unsaturated fats, you have those little kinks fold, right? So for this part, because you have that kink or that fold, it's gonna affect how that membrane can flow, right? So in the other cases, if you imagine if there were vertical lines and they kind of tangle or they move around like this, it's like, I'm almost thinking like, what's a good example? Like string, no, never mind. But here, these are able to, if you look at it on one side, because they're longer tails, and they can actually fit like this. If you guys remember, I was drawing before these molecules, I would kind of draw them like this. Like that, carbons, and you have the phosphate group, like that. And it was like their body and their tail is about the same length, right? These are carbon chains, here's the phosphate, and here's the oxygens. In the cases where you have those, that little king folding out, now you're let's say like the leg kind of kicks out this way. Now their top is small and their body heavy, right? So their body's a lot wider. When they do that, it affects the shape that how they can pack into each other. When you affect how they pack into each other, what's gonna happen is if they can pack vertically, very stacked like this into each other, you're gonna have a lot more interactions, which means it's gonna slow them down and their ability to move around. When it does that, that's gonna make it more viscous or it's gonna help them solidify, right? On the other side, it's when you have these kinks or you have the tails, that basically makes a place where they kind of can push each other away. You decrease some of the interaction, and because of that, you make it more runny, you know? So here, I think it was like, they were talking about like what do you call it? The baking grease, right? Like if it's hot, it'll be solid. The moment you it's hot, it'll be liquid. If you cool it off, then it solidifies, and it'll be that based on how saturated or unsaturated it is. It'll tell you at what temperature you need to do that switch. The other one that comes into the picture that affects us too, it's gonna be our steroids, right? Cholesterol is one of them. Steroid looks pretty similar, but they have a charged head. Here, this body isn't drawn to scale. Normally, their body's a lot smaller, so they kind of help, they affect how the thing can fold together. At this one, I really want you guys to feel comfortable with this question, which is that cholesterol, it helps with changing between fluidity and in its viscous states, right? When you add it to the rest of the phospholipid bilayer. So here it's saying that it reduces the fluidity at moderate temperatures, and then when it's cold, it hinders solid, like trying to make it from turning solid, right? So you can imagine when everything's trying to stabilize and be stable, this one is normally a little bit shorter, these tails are longer, and it makes it harder to try to make a like nice packing structure because in that area they have extra space, so the legs kind of kick in, right? So that make like it'll destabilize your membrane. And the cool thing about this is that organisms, we can regulate which molecules we have present, whether we make saturated or unsaturated based on our diet. We can also affect it like which ones we actually produce ourselves. And as you can see, some like animals like that live in the cold, they actually have taken advantage of this to actually help them withstand really cold temperatures without their cells freezing over. All right, now that we talked about what it's made of, how it behaves, how it can move around, the next part to this is talking about how it works as a barrier, right? So this is what I was telling you before. You basically have hydrophilic, hydrophobic, hydrophilic. That means that the molecules that can interact with these different regions are distinct from each other. And here what we're going to look at is how different properties of the molecules themselves affect the flow of them through this membrane itself, right? So let's get this part. Oh wait, my bad, I rushed it. Welcome back to the other part, never mind. All right, so here we're going to talk about these molecules. This table, I want you to feel really comfortable with it. I prefer it for you to know, you don't need to know these values. If I'm picking up on them on the exam, I'll probably give you that part. But I would want you to know it from this side, and the fact that this is more permeable than this, right? So I'll give you the reasons now. So for this one is, O2, because you have two oxygen molecules, equal electronegativity, they're not gonna be a polar molecule, right? So again, this up here, we're looking at the non-polar region of molecules. O2 can float around in water, right? Because it doesn't have a charge, it can actually run right through the membrane itself. So it means that it will be fused through that membrane without needing any help. It can sneak in right here, right past these guys. CO2 and 2, same thing. On the next level, you have small, uncharged, but they are polar, which is our water. Here they're kind of drawing that you have some of them who will be able to diffuse through. A lot of them are going to get, or a portion of them will get kicked back, and they're going to interact. And the interactions actually might make, like the water might stick to the hydrophilic region and won't be able to diffuse through. Same thing with what they call it carbohydrates, which is just things that have carbon and hydrogens, like glycerol, sugars and stuff. Here we're going to start seeing that. We're going to start getting the entire content of the glucose and sucrose. It's a 6-carbon ring which has positives and hydrogens kicking off of it. Those groups, now because it's more polar regions and it's a bigger molecule, it's going to have a harder time going through it. So as you can see there, this little drawing, right? Less of it diffuses, more of it actually gets kicked out. And then the guys you're not going to have any luck with trying to get through this barrier are going to be your small ions, especially if they're charged, right? And what's going to happen if you have a salt ion? It's sitting in water. It's going to build a hydration sphere, right? So now let's say you have a single calcium, your water molecules are going to basically stick around it. And now instead of being just calcium, which is being chloride, potassium, and sodium, you're going to have that molecule surrounded by water. Now technically it's going to behave like a bigger molecule, and you're not going to have any luck going through the wall, right? So it behaves as a barrier. And it's going to be affected by, again, charge and size. But remember, we need to get some of those molecules into our body. And this is where the cells have come up or adapted and created this new way of actually getting these molecules to make sure that they can come in, or getting the ones we don't want to make sure we get them out. And this is, we're going to be talking about proteins. Let me start with this one, I'll go backwards. So we're talking about proteins. This is going to go back to a little bit of what I started here today. They said there's 2 different types of proteins, right? Like I said before, we have these peripheral ones that are basically just sitting on the surface. Then we have these integral ones. And the thing about those is that they hit that hydrophobic region. For there's going to be 2 parts. One of them, the integral ones, is they go into the membrane, but they don't come out the other side. The transmembrane ones, they go all the way from one side all the way to the other. So with some of these, we have some proteins that have been adapted to carry out extra functions. Also, I'm going to go back. They go all the way through this, and they do this by changing the amino acids in that region. So what they'll do is you normally will find alpha helixes in these regions, and the alpha helix basically means that your remember your amino group and your carboxyl group, those are on the inside, and then sticking out, you'll have your R groups or your different side chains. With different side chains, you can have 4 different types of amino acids, right? Your acids, your bases, your hydrophobic, and hydrophilic. What these will do in this region, this is where they'll be putting their hydrophobic. In yellow, you can see those amino acids. Those will be your non-polar, basically R groups. That way, they'll be sticking out into the kind of like this, and what they'll interact with will be with the hydrophobic amino acids with the hydrophobic region. Now, once you get to these outer edges, here in green, that's where you can see you can find the polar ones, or the ones that are hydrophilic, and those will be able to interact with these outer parts of the membrane. Yeah. So this was the second question I put on that midterm for you guys just to guess. It was this one. It was just like, hey, throw hydrophobic amino acids in the middle, it'll be fine. Throw hydrophilic at the edges, it'll be fine. Like just by switching them out. But normally instead of just having one, what they'll do is they'll basically make like tunnels. So they'll use multiple of these, and they'll kind of put them like a straw. So you put one here, put another one here, put one here, you kind of go in a 3D circle like this, and you basically make like a pore. You can go literally right through that part of the membrane without ever touching these hydrophobic sides. And that's kind of like this little picture here is trying to show you, like you can see in here would be the pore. Technically, your molecule can come in through that green area and go through the inside. All right, so last time I told you guys talking about the different proteins, I'm like, nah, you don't have to know them, but now you do have to know. All right, so now we're going to be talking about how these proteins that sit on the membrane, they can carry out different jobs. So we have the different types. First, we're going to be talking about the transporters. Those are the ones that can carry molecules. For this one, when they're carrying the molecule, they actually spend energy to grab that molecule and bring it into the complex, right? And you can see here this little ATP, that's the energy of the cell, right? This normally undergoes a conformational change, so that protein literally, like in my head, I almost imagine like it opens up, it grabs the part that it wants, like the molecule it's gonna import, grabs it on one side, the protein refolds, allows it to shift down, and it releases it to the other side. And then the protein basically needs energy to fold back, just reset itself, to grab the next molecule. For this type, the molecules will only be moving in one direction, right? Because it literally physically grabs onto it, changes itself, lets it come inside the lung, through the membrane, and then releases to the other side. When it goes back, it has to be empty, right? For transporters too, we can also have the ones that are passive, which is kind of shown like this one here. This one, my favorite one's called the aquaphor. So that one, again, uses alpha helixes, makes like a straw through that plasma membrane. You can get one molecule of water at a time to pass through that pore, but it relies on the osmotic pressure, right? So the water will follow the sugar and the salts. Whether there's more sugar or salt on the inside or the outside of your cell, that'll tell you whether the molecule of water goes in or out. In that case, they can go both ways, in or out. Right? The next part you can find on these membranes are also enzymatic activity, which means that you'll basically anchor proteins there, and they can do different functions. Either they can cleave proteins. I think I showed you a little bit of those when I was trying to talk about the import into the ER, but they can cut proteins, they can cleave proteins, they can phosphorylate proteins, so they can do different functions. This kind of gets us into this next one, which is signal transduction, which is that they'll purposely have two different parts. And what they'll do is, on the outside, let's say when the cells are trying to communicate, there could be one cell that produces a single molecule to talk to its neighbors. This molecule then goes, finds what we call the receptor on the target cell. That second cell then grabs that. On the outside, can recognize the molecule, but on the inside, it goes either a conformational change or a phosphorylation change, which then signals to the next protein to go have an effect happen. Normally, this always, for the most part, what ends up happening is the receptor gets activated on the outside, the inside gets activated, that goes and activates normally transcription factors. That means that those will go into the nucleus and you'll change your gene expression and make new cells, or like new proteins get made. Then the other things that we can have them do is, again, trying to get information from the outside in. You can also have them do cell recognition. And this basically means that you can either have it recognize the sugars on another molecule, the shape of a protein on another molecule. Later on, I'll talk about integrants, so pretty cool, but they actually recognize each other. And that's kind of gets us into this guy, where it's like, you can actually use them to kind of link up proteins together or link up cells together. They kind of can get these proteins on two surfaces, grab onto each other. They anchor to each other. They pull each other so tight. This is how you can get this thing to happen. Right. You can get them so close and you pinch them off so tight that the membranes can merge into each other, and then you can make one single neutron, basically one bigger number together. All right. And the last one is you can also have them linked to the extracellular matrix. So remember that one has glycoproteins. In plants, we also have the cell wall, but those will be the linking spots. All right, here we're super zoomed in. We're talking about looking at the surface of an HIV virion, is that what they call it? Or basically a virus particle. So inside you have so here you have your RNA for HIV. On the outside, these will be your surface proteins. I think you guys are pretty familiar now with surface proteins because of COVID, right? You've seen the little model with the little spheres, the corona thing. So here you have receptors on the cell that it's gonna try to attach to. These two proteins are sitting on the surface. They normally are responsible for receiving the signal, right, to detect something. This is what it's getting tricked. It's using the virus is using those receptors to actually attach itself, and that's sort of get it to fuse its membrane into the cell to get it drop its content into our cells. It then tricks our machinery, uses our RNA polymerases, our DNA polymerases to make copies of itself to make new viruses. However, some people have mutations in the CR5 receptor, or it's a co-receptor. And since that's broken, this thing isn't able to anchor properly. Because it's unable to anchor properly, it's able to get tight fusion, it doesn't bring in, it can't join the membranes. And technically, you have this mutation, like if you're missing this receptor, or if you target it with like a chemical that binds that receptor itself, so you can't have virus bind to it, I think I remember one of my old friends was doing like, she was using antibodies that go bind the receptor. And if the antibody would grab on, because it was blocking the pocket, HIV couldn't stick in. So as long as you target it, you're able to block that interaction. You're not gonna let that membrane fuse. Technically, you're gonna stop the thing from being able to replicate. All right, and then the final part, I think is the last one slide for today. Yeah. Let's go back to this one. All right, and then the last thing is, we'll talk a little bit about today is these proteins. Remember, when we were talking about the ER and Golgi, we said that in the Golgi, our sugars can get sugar added to them, right? They can get glycosylated. So that happens to the proteins on the inside. It can also happen to the proteins on the surface. Going back to this drawing, you remember when we're going from the ER in the vesicles, we travel to the Golgi, the inside part of our cells, of the inside part of the vesicles, ends up being on the outside, the surface of your actual cell. As this goes inside of the Golgi, here you can have a glycosylation happening on the inside of these proteins. Then when you get to the surface, the sugar modifications you made will be on the surface cells, right? The type of sugars you glue, the way that you glue them, it's a way that your body can actually recognize where the thing is coming from. You guys look up blood types, this kind of goes a little bit into that. All right, so carbohydrates and how you're making glycoproteins. Cool. Yes. And I talked a little bit more heavily about this one earlier on, but all these proteins, one of the functions is talking about that transport. I'm not gonna go through the slide because I basically did it before. So here it was like, I was talking about they move, you have the channel proteins, they don't need energy, things can go both ways. And they're specific to, they can be a little bit more broader. The carrier proteins, they actually bind and grab onto the molecule to move it through. In that case, they know to be very specific to the molecule that they move. One of my favorite ones, I didn't have time chance to put it on there, but it's, I think it's like your sodium potassium pump. And it's super cool because what it does is it literally grabs the potassium, moves them in, grabs sodium, moves two out. Like it's literally swapping them back and forth. Yeah. All right, any questions? No? All right, cool. Happy Halloween, guys.